speaker today. He is going to present a kind of a combined structural and diffusion, as well as functional network connectivity analysis that he's been working on really for a good chunk of this summer. And Brad and I have been kind of assisting him along the way with this. And this study is going to focus on the PREDICT-HD data set that was collected worldwide, actually, between 2002 through 2017. And that data collection included structural imaging first from 1.5T scanners, and then it proceeded to start collecting diffusion data early on at 1.5T, and then eventually, as time progressed, our site started getting 3T scanners, and our data collection procedures improved, and we marched forward. And then towards the end of that study, 2014 on, we collected a pretty reasonable FBurn-like, actually, data set where we grabbed high resolution structural diffusion and functional resting state scans. And so that's kind of where we're at with this. And so I'll introduce Jordan, and we'll see what he has to say.
Sounds good. Okay. Jordan, we'll let you take it away.
Yeah, I should probably, can you hear me from there? Is that how that works?
Should be able to.
Okay.
So you guys online, can you hear Jordan?
Am I too quiet?
No, we can hear you.
Okay. Okay, thanks.
There's mics here, I think.
Oh, okay, that's what they
I
are.
think. I don't know which ones we're actually using.
Sorry about that, I was late. Okay. Well, thank you so much. Today I'm going to present my the paper we wrote. This is Neuroimaging Biomarkers for Suicidality and Huntington's Disease. Before I go into our study, I would like to quickly review, just in case anyone is unfamiliar with Huntington's disease as a pathological illness. So what is Huntington's disease? Huntington's disease is a polyglutamate disorder, or can be defined as that, also a neurodegenerative disorder, but more on that later. So normally people have a normal healthy amount of this gene on chromosome 4, but anywhere from 3 to 26 copies essentially of CAG, more on why that's a little different than 27 in a little bit. So typically these genes are relatively stable until you get to about 27, and then it's speculated that then essentially your mutability increases, and not only can you pass on these repeated copies down to your offspring, but also you are pre-manifested for HD until you essentially get to 36 repeats, and then once you get to about 40, then it's definitely HD, which is essentially a unstable CAG repeats that just keep repeating really on the chromosome. This obviously causes quite a number of issues throughout the body. This gene codes for a protein called Huntington, or as I may refer to as HTT. Essentially, one of many issues that can drive from this disorder is hair-cone loops can form in your DNA, as previous studies have shown. This can cause issues in transcription as well as translation, quite frankly, that can affect a number of issues throughout the whole entire body, but mostly we're going to be focusing on its impact in the brain. This is an autosomal dominant inheritance pattern. So essentially, you can pass on your repeated copies to your kids. You may not notice that you are passing them along until symptoms occur, and it progressively happens until death. So, from a neurological perspective, Huntington's disease is a neurodegenerative disorder. We don't exactly know how these mutated copies of CAG actually accumulate and manifest in the brain as shown. The leading theory is excitotoxicity through the HTT in the axon's body that actually it alters the action potential that the neurons produce. So essentially, but usually what we see from an imaging perspective, as you can see from my image here, that the CAG typically causes in a large caudate putamen with more in the CSF, which then shouldn't say large, but it's more altered, and then a degenerative
Huge. Say huge.
huge.
No, sorry.
Huge. Huge. I was like, I want to be technical about it because technically it's not really a cutting, but yeah. But it deteriorates the brain around the, as you can see over time. So this is a typical, in my opinion, a really good, I would say typical HD scan from UCSD. The symptoms that happen, well, obviously there's quite a number of symptoms that can occur when your brain is deteriorating over time. The most common ones are usually chorea, which is uncontrollable movements, really. Essentially, you can't, sometimes you wouldn't be able to notice that your arms were lifting up. Apathy, athetosis, cognitive decline, and dementia, personality changes from any alterations of the prefrontal cortex, depression, impulsivity, irritability, a lack of awareness that you have a disease, quite frankly, which is a number of things. And the thing I've been focusing on most in this talk is the suicide rate. So the leading cause of death in Huntington's disease is eventual aspiration pneumonia, which occurs from the deterioration of the brain itself and tissue loss, or due to a number of issues that are quite complex, suicide. And treatments for this disease, which seem to would rationally influence suicide, is some of the poor treatments for this disease itself. The clinical team that usually helps out with the care of this person as they deteriorate is usually multidisciplinary. They usually consist of a neurologist, radiologist, a genetic counselor, and a nutritionist, as well as many others. The typical drug, and more or less the only drug that's really prescribed, is tetrabenazine. That's mostly used to treat the chorea itself. However, it's at the cost of exacerbating other symptoms, namely the depression and your anxiety actually increase as a side effect from tetrabenazine. There are derivatives of tetrabenazine itself as deutetrabenazine, which then is just deuterium, and is in a place of hydrogen, which then alters the compound of the drug itself, but essentially it's relatively the same drug. These class of drugs are synaptic vesicular adenine transporters that then take the monoamines from your synaptic cleft, and for reasons that we don't quite understand, transport them back up into the presynaptic cleft itself, and then as a closed loop, it slowly kind of stops these neurotransmitters from passing through, which then in turn decreases the symptoms. There are other drugs that have been prescribed, but not quite as popular, are risperidone and naloxone. You probably recognize risperidone from schizophrenia. I'm imagining some people in here are familiar with the side effects that can occur. So it's not essentially an optimal treatment, but it could obviously end-to-end increase suicide. So so now that we've briefly gone over Huntington's disease, I want to talk about our study. So our study was essentially the data was collected from 32 sites of the PREDICT-HD data. This was essentially a little bit more than 1,100 participants. Obviously, it's changed a little bit from 2002 to 2017. And there has been a follow-up study continued on for the PREDICT-HD. We did not collect the PREVENT HD data, but it's worth mentioning. We looked at some of the data that was collected was the D confidence interval of the UCDRS, which is a 31 questionnaire of 0 to 4 self-reported scale of disability, and out of those 40 measures we looked at, it encompasses quite a lot of clinical symptoms or clinical markers such as or measures such as motor, psychiatric, cognitive functioning imaging, and we also included imaging itself. Typically, because of this disease progressive with HD itself, most studies do want to see a follow-up. So we saw follow-ups ranging from all the way up to 12 years follow-up since the visits. But mostly the mean was essentially 5 years follow-up out of this data. Again, you want to see the people that come back because as their disease progresses, the symptoms can increase as well at an exponential rate, so to speak. Next, we're going to talk about the clinical notion that we're discerning here, which is the suicidality, defined because it's a complex notion as the intentional act to end one's life. This can be measured from multiple ways. I think the way that the predict data measured this was through the Beck Depression Inventory, which is a 21-item self-report on assessment of your well-being to indicate multifaceted scales of depression. Other scales that were used but were not typically included because they're not ubiquitous was the Columbia Suicide Severity Scale, which is ideation for simple questions, which then is supposedly supposed to be used for and understood by everybody. I question that though. Here in the photo is an example of the suicidality itself, and additional measures were measured through the frontal systems behavior scale, or the Frisbee, as I usually call it. I think others should do. There's 46 questions that measures the executive functioning and other cognitive skills and abilities, such as the ones I wrote here. So we did 2 studies throughout this presentation that I will discuss. The first study was more of a statistics measure. We basically looked at the participants of the accepted suicide, tried to sort them into 3 different categories of no suicidal ideation, and whether they had a recent attempt or not. And we looked at the MRI and proximity to the assessment of suicide based on previous studies with bipolar and unipolar depression. From the predict data, from the predict sample and collections, from the predict data itself, we used 443 of the individuals in our sample, and we divided all participants into the response as I described. The demographics themselves were essentially interesting because and worth mentioning that most of the reliable statistics were Caucasian, and so I want to make that as a discretion. The second study that I will discuss that we later on was more of the imaging paper in machine learning. We used all predict HD participants with assumed fMRI data and the measure of suicide. We found 100, and of that there was 112. We again used this, the different categorization for each group, and then I will describe in that when we get to that slide what I did for that one specifically. So for the first study that we did with statistics, we looked at, as I previously discussed, the 443 people. We looked at mostly because we wanted to rule out the attempted people. We looked at the 2 groups. We used something that HD called the CAP score, which is essentially your age, which is a way that clinicians use to monitor your disease burden and Huntington's disease. So it essentially is your age times the CAGRP minus L, which is a constant, and I think it's like 30, and divided by another constant, which equals 6.59 off the top of my head. And essentially that's a scale that clinicians use to look at how your disease is progressing, and so they can relatively see how far along you are in prognosis. So we covariate that out because that's, to measure disease burden. We use ordinal logic regression because we are using ordinal data itself, and because of the type of response that we're based on the type of assessment and regression pattern we're doing, we can discern this is the J-1 categorization, which means that we can basically organize the variables from high to low and low to high, and that's sequential. In factoring in certain demographics, we were able to find multiple data here presented on the tables, as well as other clinical measures. With the imaging variables that we did run statistics on, I presented this data. I usually go for questions. I'm not going to do that this time. We also measured these out with normalized regions and used z-scores to figure out how weird something essentially was in this in the imaging markers that we used. What we found was that controlling for disease burden or CAP score, that there was no demographic differences, thankfully, between the groups with HD and without the suicidal ideation. We found that HD with suicidal ideation has a greater moral abnormality, so their motor score was not quite as great, and a worse functional capacity, which was expected. And spike equivalent cognitive speed from trails and Stroop. Really briefly, the Stroop test over here is, just in case if anybody doesn't know, is you essentially have 3 steps. You list the colors off in order, like, so in this case, the blue, red, green, then as fast as possible, then list off the words, red, green, blue, and then as fast as possible, and then we list out the colors as fast as possible. We time you out. We time to see if you can essentially differentiate between the different stimuli that you're having, that you're seeing through your eyesight itself. In a similar test, we have the trails, which is essentially you connect the dots, and we do a similar concept where instead of your natural language with the ability to discern color is taken over, instead we're looking at your spatial ability to connect dots and your ability to identify digits. So one would be like connect the dots as fast as possible, connect the dots using 1, 2, 3, and so forth. So we looked at that. We found that despite the equivalent cognitive speed, participants did perform worse on tests with executive skills, which was interesting itself, and does speak a lot to the cognitive abilities and progression of disease. And we found that HD with suicidal ideation had a much higher psychiatric scores in apathy, disinhibition, executive functioning, depressed mood, which kind of suggests that there is an underlying factor in the depression with with Huntington's disease itself. When we controlled for motor functional score, we used it and COVA, showed that no more MRI differences between HD with and without suicidal ideation was found. Into the imaging process, we did look at the resting state fMRI. Each scans were smoothed and realigned using intraline and interpolated images. We then applied the Neuromark ICA, which, as everybody here probably knows, is that it's an algorithm that's essentially used for spatially to get to more or less use as a source of reliance for spatially independent, especially independent template with time that is not independent. And we found that, and then we obviously use that to generate the static functional network connectivity. That is what we were showing here. I'm just going to briefly show that for the sake of time. We then looked at we then used spatially constrained ICA as well to find certain parameters, and then obviously, which I will discuss in the next slide, is we use the natural, not natural log. We use the negative log itself to create thresholds for the components that we found. What we found where it was.
So can I ask a question?
Absolutely, go for it.
If you go back one slide, are both a resting state fMRI and static FNCs?
So different groups, suicidal ideation and versus no suicidal ideation. Just trying to find that.
Right, but they are both resting state and both
Yes.
static FNCs.
Yeah, sorry. Yeah. They're both resting state, yeah. I can freeze it here too.
That was my question.
Gotcha. Does anybody want me to freeze it here? I want you guys, sorry.
Well, you show us the differences on the next slide.
The next slide.
Yeah.
Oh.
Wait, okay.
Right.
Oh, lad, that's weird.
That's sort of the negative one.
Yeah. So we found 84 ICA pairs that were significant. I'm going to highlight.
Is this the difference, or is this?
This is
the
the
static.
difference. This is the difference on the static.
So is it suicidality minus,
That's, I think, no.
go to the previous
Yep.
slide, sorry.
Hmm.
So is it, oh, I see, static FNC, rest, so what's, so,
I think it's just an
I think
awkward
that
space
that's
at
a
the top
mislabeling.
where it looks
I
like.
think in the bottom is the contrast you want, suicide ideation versus no suicide ideation, right?
Mhm.
They're all static
Mhm.
FNCs,
Yes.
right? So,
Yeah.
so go to the next, that's probably just one continuous
I think it's
term,
just
got
the
it,
spacing,
okay, I understand. So go to the next slide. So,
I had some issues with PowerPoint, sorry.
no, it's fine, I understand now. So is this a difference between ideation and no ideation?
Yes.
So
What's
it's
the
a,
direction, which is first?
so it's no ideation is first, and it was a rank based ANCOVA, so it was it's not a t-test. Right?
Okay,
So it's we didn't put the f-stat in here at all, just the logarithm of the p-value.
And it's because the
okay,
ordinals had to be rank based. That helps. So a t-test wouldn't be sufficient. Wasn't going to be sufficient.
Yeah, it's very skewed because it's like 10% of the population or so or less are suicidal.
I'll touch base on that in the discussion if it helps too, because there was a thing we have a sample. But yeah, any other questions before I move on?
So these edges are stronger in those who have suicidal ideation?
With HD.
or is this any difference?
It's just any difference. It's we didn't look for
these are just group
directionality.
differences unsigned. So it doesn't tell you the direction, it doesn't tell you whether which group is higher or lower.
Right, but we see they're only on the positive side.
Those are
versus
p-values.
Oh, okay.
Yeah,
Yeah.
and it's just the negative log.
So
Okay, okay, that's what I was
what you
missing.
could do is multiply this times the sign,
Mhm.
no,
But the sign of the f-stat doesn't make as much sense as the sign of the
no, the
t-stat.
sign of
Yeah.
the difference of the correlation between
That's
Yeah.
an idea.
the average of the two groups.
Yeah, that's something we could do. Easily might help with interpretation.
Yeah. See if that looks, because then you would
Yeah, I
know
know.
that,
That's why I tried to
well, at least then we would know in the actual FNC matrix, the average is higher
Okay,
or lower,
probably not.
Yeah.
which group is higher or lower in. So that might be informative.
That's why I put them
Not a t-test. It's a rank ANOVA, rank adjusted ANOVA.
Maybe I should have moved it over. Oh well. So we found 84 ICA pairs that were significant. One that I'm going to spell that I want people to pay special attention to is obviously this, the one that's 52, which is the strongest one, because that one was probably going to be discussed a little bit later in the sensory motor area. And we found the figure shows the top 24 differences between suicidal ideation and no suicidal ideation. Most finds in default network mode, which is a state one, considered to be active in its passive test. So self-explanatory. HD with suicidal ideation was not test. Was that it? I don't think I think I might have do not rest. Sorry, my eyes are a little weird. Mine's may without rest may reflect the negative inclusive thoughts themselves. So this was we also sorry about the space. So the dynamic functional activity showed 4 states. We just in case if you're wondering for the windows, we did use 30 for the grouping the windows up. We then used k-means as a way to cluster the like-minded windows themselves.
What's the length of your time series? Did you say that? Sorry if I was.
So the window
That's
size,
just
which
the
was
window
30.
Right, but the total,
I don't recall offhand.
or roughly how many windows do you get to know?
I'm trying to remember. I can get back to you on that if it helps.
Okay.
No, it's a good question.
The T
Right,
is like 2 on this, right, Jeremy? And it's
2.8 seconds.
right.
It's the Epsilon phase 3 sequence.
Okay. Yeah, we just use the defaults for the window length. It says like that.
Yeah. I was just wondering, like, the whole length of the entire time series, like how many windows you would get with.
This is 100 something. I don't remember
Yeah.
exactly.
Okay, that's fine. I was just generally.
6 minutes.
133.
Okay. Thanks, Jeremy.
Thanks. We can use k-means to group the like-minded windows together, so we can actually see the clusters themselves for as individual states, and the interpretation is based on significance. It appears as such as the correlated components, as shown here.
This is just state one, or
Yeah.
which? Okay.
Yeah, this is just state one. I just thought that was the most interesting one. So, state one is just the default network mode changes themselves. I think what was really interesting about the default mode network is that, as you can see, it's kind of it's not quite default. So that's why I included that one. State 2 is more sensory motor visual network changes that will show components as well. State 3 was the cerebellar network changes, which is also really interesting, and I'll discuss more in the discussion itself. And state 4 was intense activity in all 4 networks, and the at Neuromark themselves will be discussed because it looked like it kind of. I mean, I'll discuss it in more discussion as well.
Which state was that one you're showing?
It's just
Which
state
one? This is state 1.
That's state 1. Okay.
I know it looks really.
I don't understand why you're showing both of these FNCs, because I can't really see any difference between.
He's gonna go. He has the group differences on the next 4 slides.
I had them. I thought it'd be better to show more data as opposed to
You
less.
need to work on your FNC Zen, Cyrus.
Yeah.
It's obvious that there are differences.
One right
The negative correlations.
I need
and
to get
the
glasses.
negative
Yeah.
correlation. I do too, kind of. The negative parts, if you can see from the blue.
No, you can see, right?
You can
This
see
one is
some.
much more blue
That one's blue.
than this one, and this one is more yellow than that one.
Yeah, we have group differences.
Sorry. Maybe a little bit, yeah.
You could also just take the difference and show
Yeah.
images.
That's what we should do. Yeah.
Yeah.
Yeah, that's probably, yeah.
Magical power of subtraction.
Yeah, that would have been great. Okay, well that's a much better idea. Kudos. So essentially this is the first of state 1, the default mode of the dynamic FNC connectogram itself. Again, we can see the 52 components themselves, but we also see the cognitive control activates with all of their networks. So it's probably that there is it probably underlies the issue of I mean, I'll touch on this a little bit more in discussion, but it does seem like there is an executive functioning issue that HD is affecting with depression. So I think that's why this one, that's the thing I'm hoping that people will take from this connectogram. The second state 2.
I have a question.
Yeah.
Yeah. Here, is a group difference analysis?
These are group differences again, and it's just the p-values again. So it's the negative log of
Is
the
it
p-value.
significant p-values?
Yeah, these are
Yeah.
thresholded, yeah.
Threshold, yeah.
It's not signed though. There's no
Yeah,
direction.
they're not signed like we normally do for the
Positive
t-tests.
or
So I
negative.
want to know
Because it's
what is
correlation.
significant
Yeah.
connections.
Yeah, and so this was quite aggressively
Just
thresholded because we got really low p-values even after correction for the dFNC. So this was like a huge
Low
threshold.
p-values, 65 logarithm.
That's
It shows that these connections were significant. Only these
Yeah.
these were significant between the
Yeah,
groups.
this is just aggressively thresholded, I think, like 1 times 10 to the negative 10 or something like really tiny.
It says 65 on the.
It's because it's the logarithm.
log.
Oh,
So
Again, we did the common trick to make
Mhm.
like brighter is more significant, but then we did thresholded again because there were just so many.
So, yeah, this is a default mode network within itself. This is so what's the comparison here?
This is the group differences in state 1, cell-wise.
So a p-value with 65 zeros.
Yeah, something huge. I mean, this is after correction as well.
So go back to the previous
Slide.
So it's the difference between these two.
I believe so. If this is state 1,
yeah, state one between two groups.
and it's default mode within itself. Okay. That's-
look at what we said.
So this, this,
mhm.
So
right?
several different.
I don't know. I'm not buying it. I'm not buying it.
Maybe that's a different state then, right? So
Could be.
Could be, yeah.
Yeah, I think what you
that seems so
Vastly different, yeah.
different.
Yeah, I think what you said
given
is state
this
3.
Yeah, that looks like
And the group difference is ideation, right? Again,
Yes, and with attempt, obviously too.
and with what?
With attempt.
And attempt, okay.
Yeah, so you put state 3 in your
I think I did.
first
Yeah.
slide.
Okay.
Apologies, I misspoke.
So how does state 1 look when you look at? I look at it just to see.
It
Visually, you should
looks
see.
it looks much greener, like green.
Let's see.
It makes me wonder if there's an outlier in one of the groups.
I mean, I can see the differences. I'm looking at the default mode right now in state 1. Those differences are pretty obvious. There's just like the 2 cells or connections that show for that state. I mean, for your paper, we'll have all the states in there as well. But this is just
Anyway,
for the
it's just related.
presentation.
Yeah. No,
You got the
that's
gist.
that's good advice. Yeah.
And just one more thing, is GLM analysis or two-sample, which kind of
It's again a rank adjusted ANOVA.
Rank
So
ANOVA.
so we're doing and we were controlled for. You mentioned this as part of study 1. It's actually study 2. That we control for the motor score. So that's why we needed to do an ANOVA. And ANOVA technically.
T-test wouldn't have worked on that. So.
And the rank adjustments for the multiple testing adjustment.
Thank you.
Sorry. So yeah. So the cognitive control activates all the networks. I'm going to try to breeze through these time-wise. Here is state 2 with the sensory, motor, visual connectivity.
Is it the same rough scale
Yeah.
for the colors?
The threshold's the same.
Yeah.
Yeah.
And then I'm going to go to the third state if everybody's done with this one, which is this stuff.
Right.
Again, super.
So
high.
this was the one that he was actually showing, I believe. This state was the one that he was
Yeah.
showing.
Can you go back to that?
Yeah, the original one. Sorry.
That's even higher.
So if you look at the cerebellar cells, it should see some stuff in the bottom right.
I don't think.
To here?
Yeah, this is the
I can see
the mean doesn't look that different to me, but
Slightly.
yeah, I mean I can
It
see
looks
a
like you might have
difference.
plotted the same for each group as well, because it
But
looks
you're
like
doing the test you're doing here is the same ranked
you plotted exactly the same.
ranked ANOVA
So-
or whatever. It's the same
I don't
test.
think it's the same.
Oh, it's not the same. And it's
This is more intense than this.
That's subcortical.
This is some differences where it's subcortical. It's
Cerebellum's the bottom. The
Right.
bottom.
There's some differences, but it's not that drastic.
How many subjects did you have?
You should give a look at Brad's computer screen and stuff. I can see his.
That's what I've got to pull up, but
You gotta each one of those. I do too. I
yeah,
think
we just
it-
gotta make
You
sure.
should do
Jordan,
a
that you're-
white, 0. I like the,
Like-
then,
White to red.
red to blue with white in the middle. I think it makes everything
Sure.
pop better. I have to see with the green.
How many subjects did you have again?
In which in side 2, that would have been 100.
Yeah, the
Yeah,
white. I like the white
More
ones.
than
Yeah.
133, I think. Let me 112 that we used for this one.
Yeah, that seems like a really low p-value.
Something's going on. Yeah, it's worth double checking.
Oh, that looks better. Yeah. Wait, okay. Thank you. What's that?
When you do these tests,
Yeah.
you set them up between all the windows for each participant in a particular group, right? That's all we did. So it's, I don't know how it's low, but it's you get more windows for each participant. So seeing lower p-values.
So the
yeah,
p-values aren't exactly on those averaged. So what you're showing is the average state difference.
Right, you don't run it.
P-values are being calculated from each
from all the
individual.
windows, right?
Oh,
You don't,
Okay, so that makes
you
sense,
have like
actually, then it
100
wouldn't
right,
show-
over 100 windows
Right.
times subjects.
because you don't run the test, you don't compute the mean and then run the test on just the mean. So you can do that. But
Right.
that would be a t-test only.
Okay.
Right, so.
Yeah.
The thing is, I think I had the same thing that seems to have suggested to take mean of the windows per subject and then.
Oh, are you including all the time points in the?
Yeah.
Oh, that's the problem.
Yeah.
Those are
You should
dependent.
take your
Yeah.
average.
So you have to. I think it's
subject.
not valid because you're not accounting for the dependencies in the tests you're doing. You're treating them all as if they were independent subjects.
Mm-hmm.
So it's like you have 112 times the number of time points subjects, which is why it looks so significant. So it's like a UK Biobank, thing, but they're not independent, and so that makes them have a very low variance too, which
I
right?
see.
So the right way to do that would be to average
Mean per subject.
the
Yeah.
all of the time points within each state
Mhm.
within subject, and then test between groups that way.
It's kind of like fixed and random effects, right?
Yeah, it's sort of, yeah.
That makes
Same as
sense.
what Jared
Yeah.
said.
Yeah, that helps a lot and explains why we got such a low p-value, because I was a little concerned about that. Okay. It was like 0.002.
Usually there's a reason for such a low p-value, so yeah.
Okay, that's great to know, because I was like, that seems really too good to be true. It was, I think it was off the top of my head, 0.000.
All right, let's move on.
Okay, sorry.
Well, let's talk a little more about this then.
Dynamic, so we looked at the intense hyperactivity and state 4 itself. So I'm gonna go through that. So our results out of study 2 was the connectivity was different between ADHD and the suicide ideation. Again, most studies findings were in cognitive control circuits between different components themselves, and but most of these components do have certain domains. There was obviously the visual domain with the cognitive control. There was a placentral gyrus, I think it was the most like it was auditory circuit with the structural area of the postcentral gyrus, which is essentially close to the caudate putamen itself. So that does seem really interesting as to why that would be affected. So here are the results. There is previous literature that does suggest that then the hyperfixation on this or the intensity in the cerebellar domains have functional uses as a social emotional processing and sense of self, and how people in autism spectrum disorder associate with identity. So we did hypothesize that could be possibly why we're seeing so much components in this cerebellar area. Ugh. My food is just digesting, so this is great. Good timing, I know, right? Blood pressure's dropping. Discussion, okay, so our primary takeaway was the findings are consistent with essentially the previous studies that are bipolar and have bipolar and unipolar depression themselves. There are some slight differences, which could be attributed to HD. The questions are we wondering is why is there not some cortical measures from our typical Neuromark area, and why not sensory motor areas themselves? One of the huge limitations that we had in this study was the small sample size. Part of this was due to over time from 2002 to 2017. We went from a 1.5 Tesla scanner all the way up to, I think I did see recently, that could be Prevent 7, but we didn't use Prevent. So I think that took away a lot of the data that we had in this database itself. Another thing is it was hard to discern suicide measurement, especially from clinical from rating skills present. I do question some of the precision of what these concepts are actually measuring. Internalization, and in turn, how that works, internalization is also problematic. Also, we did notice that, I didn't include the figure in this presentation, but we did have a strange deviation for when somebody attempted suicide, how they had a slight difference in their measures. So that is something else that does seem kind of fishy and weird. The level of type of suicidality, we don't know that this suicidality could be caused by unipolar depression, multiple diagnosis of bipolar, or even if the type of depression is just due to low impulsivity itself. Also, this is more just me being nitpicky a little bit about the idea of a domain and a component itself. Is I can't really I do kind of wonder if how I can say that the domains that we found from HD are actually similar. Are they being treated the exact same as bipolar and unipolar depression, as they are very complex diseases that are very different? And so I do kind of question the areas of components that we found. Going forward, I think it would be really good for we measure the components correctly, probably, but we also test for other symptoms like OCD itself. That's another clinical symptom that has been present in Huntington's disease itself. I think chorea has been well explored. I do think there's less so exploration of the symptom of compulsive behavior, and I would even think irrationality and irritability should be explored as well. I think joint ICA could be applied to this data set as well. I do think obviously a larger sample size would be much easier, and there's more methods that we could apply if that was the case. And because it's only 20 years, a mean of what followed 12 years for some individuals, I do think that a longitudinal study will be in need, just to see how the disease progresses over time, because this is just a simple window, a tiny, when we're seeing them, it's almost like an instantaneous assessment, really, of their disease burden. And it would be really great if we had more follow-ups over longer periods of time than just 5 years, since this disease does progress for a while before death. And seeing how it changes is, I think, important. I do think that the prediction, the previous studies, as I showed here, have used prediction accuracy with clinical variables and machine learning. I do think that a similar approach could be used for this Huntington's depression that we're looking at. I do think that could also be used to predict suicidality. And I think more imaging features like PET could be implemented as well, not just fMRI. And because I am transitioning to look more into immunology and the microbiome in this past year, I am kind of curious that if I was to recollect the data all over again, if I could assess for certain bacterial specimens, hopefully when RNA sequencing is more progressed and there's less of a dark microbiome, because there are some microbes that do actually possess the HTT gene itself, and I think that could influence measurements as well, and as well as the brain interaction. Thank you so much. I hope this didn't take too much time, and let me know if there's any questions.
That's great. I think very cool stuff. So what other data are you planning to look at in this?
Okay, so in the next coming year, I'm going to be learning some immunostaining techniques, and one of my hopes would be if I ever do get to that step, because I do have R set up on the computers. I've looked at how they assess it, and it does seem like I could.
RSEP, what was that?
Oh, sorry. I could. It does seem like they have. I would like to take microbiome, the microbiome of participants and new subjects, if I could, and then see how that incorporates. I think that's something I'm really interested in.
Okay. This is and are you? Does everybody have microbiome or most?
Most HGD, yeah. No, we all should have a.
No. I mean, did you collect it for everybody?
They did not in predict. That's why I would like to I think it's kind of important. There's been other literature that has just looked at as an animal model. They've just looked at bacteria with Huntington's, which is interesting from my thought, because some bacteria have an SOS response where they just flood their genome. They're talking about adding it also, I think, for Castellan predict, but I think it'll be huge, specifically because some bacteria they have an SOS response, which essentially is like they flood their plasmid with like a whole. They just like go into hyperdrive with like a replicating and throw out any proofreading, so they can essentially make errors. And I think if you do accidentally make more CAG repeats in that SOS response, then you're going to have even more CAG of that gene expressed. So there's been previous studies that have looked into that. And I think that alone, with just how.
So where will you get the microbiome data?
So the assay itself, I think I would have to collect it from scratch, because I don't think it's in this database. I think I'd have to do it over again. But I think that's okay, because a lot of the older data is like 1.5 Teslas. So it needs to be collected anyway, really, to use your ideas.
You would be looking at microbiome and imaging together?
Yes.
Oh, okay.
Because it would need to, because right now most of this predict stuff is like 1.5.
But if you don't have the microbiome, how will you get the you would go back to the subjects?
Yeah.
Yeah. Not the same subjects. I have to recruit new subjects. So I have to start all over.
Oh, okay. So you'd have to do scanning.
You mean
again,
for
too.
this one?
Yeah.
You mean for what we're doing follow up? For Castle,
Yeah.
it's being added on, I think.
Oh, okay. This is B for Catacil. Okay, got it. Yeah, sorry.
They're all also doing HD, but I think that's because Prevent is still going on.
So Catacil's already ongoing, and so if you get it in a subset of those folks, you can look at the relationship. Yeah.
Yes.
Because that's been starting to be looked at for some studies of AD, I know. So
Yeah.
Yeah.
So we're just starting. So it'd be added on because we're doing visit 2, I think, this fall, if that helps.
Okay.
So
Okay.
then Prevent is kind of going through its next cycle.
Yeah, there's a couple papers where we looked at it in alcohol and also in schizophrenia, microbiome and FNC. So you can check those, maybe be helpful.
I would love that, yeah.
Yeah, cool. All right, well, thank you. Any other questions for Jordan? Thanks for presenting. All right, thank you, everybody.
I was observing the last year, right? So I tell you,